Now, gene expression also happens when proteins are synthesized. So, post translation, there are other methods used to make sure that gene expression is happening, is controlling how things, how the proteins behave. So, when we have a nascent polypeptide, the nascent polypeptide is normally never functional. There have to be numerous other modifications before this nascent polypeptide enzyme can be functional. And this involves a lot of things. And here are a couple examples. So the first is once this polypeptide is formed, somehow the cell has to be sure that the specific 3D folding is accomplished properly. So this entire polypeptide has to be folded in a correct position, in a correct manner, and that's done through a family of proteins called the chaperones. The chaperones are like shepherds, because that's my stupid analogy right there, shepherds. But they're just making sure, well, shepherds because they're taking care of sheep, but the chaperone, on the other hand, is taking care of the amino acid. It's making sure that it's folded in the right shape. If it's folded in the right shape, it's called the native formation, or sorry, the native conformation. If it's not folded in the right form, it's the misfolded protein. It's called the non-native conformation, and that's the two key terms you should memorize. And I should have had them read and kind of like colorized, but whatever. Let's just move on. And we have examples. An example of these would be of the use of chaperones would be in the nucleosome assembly. And we understand how important it is. We know the nucleosomes are. We have 8 histines that are wrapped around with the linear DNA, and that just helps us conserve space and properly, properly save or conserve DNA. And that's done with the help of chaperones. The chaperones assist the entire folding of the chromosomes. And we also have another modification, covalent modifications. Covalent modifications occur, numerous of them can happen once this nascent polypeptide is made. There are numerous covalent changes that are made. And here are a couple of examples. The first is it can be acylated. And acylated just means that an acyl group is attached. And normally it's attached on the N terminus of the amino acid. So the lysine amino acid inside the histines will be acylated, and this will cause a positive charge to be decreased. And because of this, the DNA can loosen up more, and this allows transcription to happen. And this is a changing from heterochromatin to euchromatin, if you notice that. And we have formulated. So formulated might bring some bells already. So remember the FMET from prokaryotic translation. The first amino acid is the FMET. It's the methionine. So the formulated methionine is the first amino acid inside the translation of the prokaryote, whereas the eukaryote just has the methionine. So formulated just means that if we have a COH group addition somewhere on the molecule, sulfated is very similar to formulated in the sense that you're just having a SO4, a sulfate group just being attached. So a tyrosine amino acid can be sulfated if we have a SO4 2 minus attached to it. We also have an alkylated, what alkyl group is just attached to it. And this is a post-translation modification as well. We also have phosphorylated. With phosphorylated just means that we have a phosphate group that's attached, and this can happen to amino acids. And we also have proteins that can start linked to one another as well. So these are just some examples of covalent modification, and really organic chemistry will cover a lot of